Hello and welcome back to EASD TV. The prize lectures are a very prestigious part of the EASD programme. They're awarded every year and their recipients are really the top, the creme de la creme of EASD members. And I'm delighted to welcome the winner of the Albert Renold Prize Award. And the Albert Renold lecture is about basic science. And with me I have Laurie Sussel from the University of Colorado in Denver. And Laurie, firstly, how did you feel to be awarded this prize?
This was a very great honor, not only to be awarded such a prestigious prize. Albert Renold was a very passionate scientist, but to also be nominated by your colleagues and peers for the award is very special.
And tell me your subject area.
So we're really interested in how the islet cells develop during fetal development, and also understanding the basic biology behind how they function and how they secrete insulin, make insulin and secrete insulin. And the hopes there are the more we understand about the basic biology is the more we can develop cures for type 1 diabetes and for fixing those beta cells in type 2 diabetes.
Now, I know diabetologists kind of feel that they know everything there is to know about beta cells, but actually that's not true at all, is it? What are the big gaps in our knowledge?
So the big gaps are, we used to think that, especially in type 1 diabetes, that the cells were destroyed by the immune system, but we actually know that now they're not actually destroyed and they can convert to other cell types, or they become immature, so they go into hibernation. And we're hoping if we study the biology of that, we can reanimate those cells or remake them into insulin-producing cells once the disease has started.
That's a fascinating area because we know that there are factors, I'm simplifying wildly here, I know, but there are factors that prompt cell differentiation. And what you're saying is you need a kind of Cinderella factor, or Sleeping Beauty factor, I should say, to come and wake them up again.
Yeah, the very interesting thing is that the same factors that are involved in developing a beta cell during embryogenesis or fetal development are the same factors that are needed throughout life to keep those cells being functional. And what I think we've learned is that there's a lot of plasticity in the islet cells that we didn't know existed. And so they can convert to other cell types. So an insulin-producing cell can become a glucagon-producing cell. That's not what we want to happen, but it's the way I think the cell can either evade the immune system or evade the stress that's associated with, say, type 2 diabetes.
And this is important because we're already moving into a phase of beta cells which are engineered to be transplanted into humans.
It's a really exciting time. It's been 20 years of research doing basic biology and using mostly rodent models to understand the signals and the transcription factors that are actually needed to take an embryonic stem cell or an induced pluripotent cell and differentiate it into a functional insulin-producing cells. And we're actually already in phase 2 clinical trials, which none of us would have predicted would have happened even 5 years ago. So the field is moving very quickly, and it's very, very exciting.
And why did you originally get into this field? What was it that I'm always interested in scientists' origin stories, if you like. What made you go into this area?
It's a very interesting system in that a very few number of cell types actually evolve or form from a common progenitor. And so it's a system that you can actually take apart. And it seemed to me that was quite simple and I was going to be able to solve this in a couple of years, but it hasn't happened. We're still figuring out how complex it is. And every discovery we make, we figure out we didn't know something. So I think it's just one of those fields that not only at a basic level is very interesting, but then in the long run, we're going to be able to actually help treat and understand diabetes.
It's fabulous work. I know that you will be as fascinated as I am if you go and have a look at that lecture, which of course you can see online. Laurie, thank you so much for being with us, and we'll be back with you soon.